Hello, I am Eranthi Prasadika Kodituaku, and I'm going to present the research findings on fossil diatom assemblages and pyrite formation of the post-glacial stage and the Holocene epoch in Fujii area, Okayama Plain, Japan. Let's move to the introduction. So when we consider diatoms, diatoms belong to a class of microscopic unicellular algae living either in freshwater, seawater, or in brackish water. So this diatom assemblage analysis provides accurate salinity condition data. Also, diatoms will deposit in sediment when the sea level rises, and by examining the fossil diatom assemblage, we can get an accurate idea about the past environmental condition of a particular area. Along with that, the pyrite concentration of the sediment samples also provides information on paleo-environmental changes. So by coupling this pyrite concentration and the carbon-14 dating with this fossil diatom data, it is possible to analyze the changes in the sedimentary environment brought on by sea level fluctuations. Moreover, future climate changes, also sea level fluctuations, could be predicted using this knowledge. So, the objective of this study was to use the diatom assemblage and the pyrite formation to examine the sedimentary environment of Fujii Okayama Plain in Japan. So let's move to the methodology. So here you can see the Okayama Plain where we have found this sampling site. And also here you can see clearly see the sampling site, and the southern part of this site faces the Inland Sea, which contains several small islands belong to Okayama. So let's talk about the sampling and methodology. So at the first, a sediment sample was collected from a borehole in the Fujii area, Okayama Plain, and 4-meter core sample was used, and also samples from 1.78 meters to 1.95 meters and 2.2 to 2.4 meters were used for the diatom analysis, pyrite sulfur analysis, and also for carbon-14 dating. As well, the radiocarbon age measurements were carried out at the Department of Geography, Hiroshima University. Also, the laboratory work for soil samples was carried out at the Department of Botany, Hiroshima University. And the mounted diatom samples were checked by polarizing microscope. Also, the laboratory procedure for pyrite analysis was modified by Professor Katupotha following the procedures described by Berner, 1979. Then let's talk about the results and discussion. First of all, I would like to present the pyrite sulfur analysis and carbon-14 dating results. So as shown in this figure, high pyrite concentration was shown at 1.78 to 1.95 depth. Since pyrite is mostly found in brackish water and saline water environment than in freshwater environments, high pyrite concentration in this sampling depth suggests that during that time, this area has been submerged in saline water. To confirm this theory, actually most marine diatoms were also found in this depth. Also, carbon-14 dating of the 2.2 to 2.4 meter sediment depth sample was revealed to be 11,035 years before present, and the pyrite concentration gradually lowered to that point, suggesting that the sea level rise during that time period. So here it shows the stratigraphic profile of the boring. And based on the post-glacial deposits, the constituents of the bore identified as basal gravel to fine sandy layers. So let's talk about the diatom assemblage. Actually, in this study, it has been identified several species of diatoms, more than 100 species. But in this table, we have summarized some of the fossil diatoms. So here you can see it has been identified a lot of marine diatoms, as well some freshwater diatoms, and also some brackish water diatoms. So as an example, in this study, high pyrite concentration was recorded at the 1.78 to 1.95 meter depth. Along with that, in the same depth, it has been identified several marine diatoms, such as Melosira species. So this Melosira species was recorded at the depth of 1.85 meters, and this depth belongs to this particular area. So this presence of marine diatoms together with the high pyrite concentration confirms that this depth or this area was covered by sea water at 6,745 years before present. So let's move to the conclusion. So the pyrite analysis indicates that they are mainly terrestrial in origin. Also, 11,035 years before present carbon-14 data coincides with the last glacial period, as well 6,745 years before present carbon-14 data coincides with the Holocene stage. Also, this high pyrite concentration in the sampling depth suggests that this area has been submerged in saline water from the early Holocene to mid-Holocene. So the analysis of fossil diatom assemblages also reveal that the species belong to freshwater, brackish water, marine, planktonic, benthonic, as also neritic and oceanic, etc., and can be correlated with the results of the pyrite concentration accordingly. Also, this study recognized that the indicators such as pyrite concentration, microfossil diatoms, as well the carbon-14 dating are important as sea level indicators to study sea level changes in coastal areas. So this study was done as a part of the doctoral study of Professor Jinadasa Katupota. So we would like to acknowledge the Department of Geography and the Department of Botany of the Hiroshima University. Here are some of the references of this study. And with that, we have come to the end of the presentation. Thank you so much.